All right, guys. Hey, this is Mr. London. So today we're just going to be going over the mitosis notes. I'm going to try to make this as quick as possible. So when we talk about cell division, we're really talking about there's two different processes. There's one for mitosis. One for all the body cells, and that's called mitosis. So mitosis is the kind of cell division that's used to make new bone cells, new skin cells, muscle cells, all the cells in your body except reproductive cells. Okay, so reproductive cells like sperm and egg are made through a different process. But today we're going to be talking about mitosis, which makes body cells. Okay, let's move on. So why do cells divide? They divide for reproduction, and I don't mean making sperm and egg. I mean we all started as one cell after we were conceived, and over the course of 9 months, we developed into a 7-pound baby. But it didn't stop there. Your cells kept dividing, and they keep dividing as you grow into adulthood. And then it continues. Your body's constantly repairing itself, not only if you have an injury or a broken bone, but also every so often your cells die and they need to be replaced. So the cell cycle just refers to the normal life cycle of a cell, and it includes two parts. There's mitosis, when it's actually dividing, and then there's interphase, which is the part of the life cycle, the cell cycle, in between cell division. And as you can see, interphase is pretty much 90% of a cell's life. Okay, so that's when it's growing, resting, doing its everyday cell things. Okay, now the 90% is just an average. Some cells divide much more quickly, like your intestinal cells, your heart. Well, anyway, and some cells don't divide very often at all, like your brain cells. They really don't divide very often at all, which is why you have to take care of your brain. So again, interphase is that time when the cell does its everyday thing, everyday functions when it's not dividing. The most important thing that happens during interphase is if it gets the signal to go ahead and divide, it needs to replicate its DNA before it divides to ensure that the daughter cells have the same amount of chromosomes as the original parent cell. Okay, for example, humans have 46 chromosomes, so it's really important that to make daughter cells or new cells with 46 chromosomes, those chromosomes have to duplicate themselves so that when they divide, the daughter cells also have 46. Otherwise, the daughter cells would only have 23, and if they divided again, they'd only have 11 and a half, and then so on. So again, before it divides, it needs to replicate its DNA. So what are the parts of a chromosome? So a chromosome is, again, it's made of DNA and some proteins, but a chromosome is again, genetic material that we inherit from our parents. So we get 23 from our father and 23 from our mother. And it's basically DNA that's condensed into a tight little package. Okay? And this middle thing, this green dot represents the centromere, which holds the two sides of the chromosome together. Okay? And the two sides of the chromosome are called chromatids. Okay? So after DNA replication during interphase, the chromosome looks like an X, and it has two chromatids. You've got one here, and you've got another chromatid here on the right side. Okay, and they're just mirror copies of each other, connected by this green dot called the centromere. So again, this whole thing is called a chromosome. Let's go back. This whole thing is called a chromosome. This middle part is the centromere. We've got one chromatid here. We've got another chromatid here. This purple dot just represents a segment of DNA that codes for a trait, and that's a gene. So this could be one gene, for example, for whether your hair is curly, straight, or wavy. There could be another segment of DNA up here for another gene, maybe for the color of your hair. So every different spot on the chromosome is a different segment of DNA, and it's a different gene that codes for a different trait. So again, chromosomes are made of DNA mostly, but there is some protein that helps package it into um, that tight little shape that we looked at on the previous slide. So what again? Chromosomes are genetic information because they're made of DNA and they're passed from parents to offspring. And humans have 46 chromosomes. Different species have different numbers of chromosomes. And after a chromosome replicates, it has two identical sister chromatids. Okay, that's why it looks like an X. Um, so, what does that mean? So, before DNA replication, every chromosome doesn't look like an X. It actually looks like a number one. Okay, because before DNA replication, every chromosome only has one chromatid. Okay, it only has one chromatid. So again, this is a chromosome with one chromatid. The part in the middle is called the centromere. So this just represents two chromosomes. Okay, let's pretend the purple one is the one you inherit from your mother, and the green one is the one you inherit from your father. Okay, if your cell gets a signal to go ahead and divide, it needs to replicate its DNA ahead of cell division during interphase. So what's it going to do? Each chromosome is going to grow another chromatid so it has that X shape like we see here on the right. Okay? So now our chromosome from our mother has two chromatids. Here's one and here's another. And our chromosome from dad has two chromatids because again, they've made mirror copies of each other during interphase. Okay? So we actually have 23 pairs of chromosomes like this in all our cells, all our body cells. Okay, 23 pairs, 23 from mom, 23 from dad. The only cells in our body that don't have 23 pairs are reproductive cells, like I mentioned earlier. So again, DNA is found in the nucleus of eukaryotic cells. It's condensed into that tight little package known as a chromosome, especially when the cell is dividing. Um, it's condensed into chromosomes. When it's not dividing, it's actually all spread out and uncoiled. So the whole point of mitosis, so we talked about interphase, that's the phase in between cell division when the cell's replicating its DNA. When it divides, it enters mitosis. And the whole point of mitosis, this is the key point, is to create two identical daughter cells with the same number of chromosomes as the parent cell. For example, here's a simplified cell with only two chromosomes. Okay? Notice how our parent cell, the chromosomes don't look like an X. They actually look like a 1 because they haven't replicated their DNA yet. So we've got two chromosomes with one, each has only one chromatid. If they get the signal to go ahead and divide, they're going to replicate their DNA. And now we've got two chromosomes still only two, but they have each chromosome has two chromatids. Okay? So we've got two chromatids here in the blue and two chromatids here in the red. But that still only counts as two chromosomes. Okay? The way you count the number of chromosomes is you count the centromere. So I've got one centromere here. That's the little thing in the middle. One centromere here. That's two chromosomes. One centromere here. One centromere here. That's two chromosomes. Okay? But they're basically again, already replicated. So now that they're ready to divide, they enter mitosis and those two chromatids separate. So one red chromatid goes into this daughter cell, one blue chromatid goes into this daughter cell, and the other two chromatids go in the other daughter cell. So we end up with two daughter cells that are identical to our original cell. Okay, with two chromosomes in each one. So mitosis, even though it's like a continuous process, we break it down into 4 phases. And it helps to know the names of the phases just because it helps us remember what happens during the phases. So it's not really important that it's not really important about the name. It's more about what you know, that you know what happens during mitosis, and you're able to put things in order. Okay, so it does help to know the names, and the way you remember the order of the names is PMAT, PMAT. So prophase is first, and during prophase, you're basically getting ready for cell division. So the first thing that happens is the nuclear envelope around the nucleus starts to break down. So notice how we've got that dotted line, meaning that it's kind of dissolving or disappearing. And that's going to, now our chromosomes start to condense because during interphase, the chromosomes are kind of uncoiled and they just look like spaghetti. All right, they're all uncoiled and mixed together. During prophase, they condense and they become visible, and we can see here they look like an X. Okay, so we've got 4 chromosomes in the nucleus here, 4 chromosomes in the nucleus here. Again, they're condensed, they're visible. Our nuclear envelope around the nucleus is disappearing, and our spindle fiber is forming. The spindle fiber is sometimes known as the mitotic spindle. Basically, it's just a bunch of protein fibers or protein ropes, I guess you could say, that are going to grab onto our chromosomes and basically line them up where they need and bring them where they need to go in order to divide later. So these thin orange lines here and these thin blue lines here represent the mitotic spindle. I'm sorry, we'll call them the spindle fibers. So how do we remember that prophase comes first? The prefix pro means first. How do we remember that metaphase? What happens during metaphase? The chromosomes line up in the middle of the cell. So metaphase starts with an M, so does middle. So basically what happens, you see these spindle fibers, these blue lines, they basically attach to the centromere in the middle of each chromosome. So they're attached to the middle of the chromosome, and they line up the chromosomes in the middle of the cell. Then during anaphase, those spindle fibers are going to rip the chromosomes apart. So we've got one chromatid. So again, we've got our spindle fibers connecting to the middle of each chromosome. That's called the centromere, right? That happened during metaphase. Now during anaphase, the spindle fibers rip the chromosomes apart. And remember, each of those chromosomes had 2 chromatids. Now those chromatids are separated, and they're getting pulled to opposite sides of the cell. How do you remember what happens during anaphase? Anaphase starts with an A, and the chromatids are moving away from each other. Away starts with an A. And they're getting pulled to opposite sides. And then finally, we got telophase is the last step. This is when the nucleus actually divides. So we've already got our chromatids on opposite sides of the cell. Now we're just going to form a nucleus, so that nuclear envelope, that nuclear membrane that goes around the nucleus reforms. That's represented by this dotted line right there. Our spindle fibers, we don't need them anymore, so they start to disappear. And again, at this point, cell division is not quite finished, but mitosis is done. I'll explain what I mean by that in a second. But yeah, if you want to just abbreviate what telophase is, it's when the nucleus divides. Okay. How do you remember that telophase comes last, aside from the PMAT thing? T comes after in the alphabet after P, M, and A. So it comes last because T is later in the alphabet. But during telophase, you also have this thing that's called cytokinesis happen. And that's basically when the cells actually physically break apart. So we've divided our nucleus during telophase. Now the rest of the cell has to become separate. So this is division of the cytoplasm. Remember, the liquid part of the cell outside the nucleus is called the cytoplasm. So this is basically when the cells just kind of pinch apart. In animal cells, it's a little simpler. You just have this little ring that forms between the two dividing cells, and it just pinches them apart. Okay? Again, this is cytokinesis. In plant cells, there's just an extra step because plant cells also have a cell wall. This little cell plate forms between them also, and that develops into the cell wall that separates the two plant cells. So here's just a picture taken with microscopes. This is prophase, kind of like early prophase. You can see the chromosomes. They're still kind of coiled together, but they're visible. They're just tangled. This is metaphase right here because they're in the middle. And this is anaphase because the chromosomes, again, these blue things are moving apart. The thin red hair-like strands represent the mitotic spindle. And we've got telophase here. We've got a nucleus here and a nucleus here. Again, these are just taken with a microscope. Basically, during interphase, you can the nucleus is still visible. So during interphase, you can't really make out any chromosomes because the DNA is just all mixed together like spaghetti. So this would be interphase right here. This would be interphase. If you want to know, the dark little spot in the nucleus is called the nucleolus. That's where ribosomes are made. Here's one in metaphase. This one's in anaphase. This one looks like it's about to start telophase right here. You've got the two nuclei, but their cytokinesis hasn't really finished yet. Cytokinesis actually happens during telophase, but here again, you can't see a cell wall in between them, so it's not. It must be early telophase. This one is looks like it's somewhere in between prophase and metaphase. Again, it's a continuous process, but to help explain it, we break it up into 4 stages. All right, great. I'm going to stop there to keep this video short, and you can always replay it. And remember to turn on subtitles if that helps.